So thanks, Judy, for the kind introduction. And yeah, this is my first time at a Women in Data Science workshop, so I don't really know what to expect. And unfortunately, because I'm in Zurich, I wasn't able to attend the previous talks, but I'm sure they have been amazing. And it's probably a bit of a stretch now, but indeed, time series is a good transition because in fact, I'm also dealing with time series. I'm not necessarily like forecast or predict them, but this is also like some cognitive neuroscientists would also do that. But I rather basically look in these time series to understand where we find activation in the brain in response to some cognitive tasks. Yeah, so my talk today will be about AI and neuroscience and combine real-time brain imaging and machine learning. So maybe first, because I know most of you won't have a background in cognitive neuroscience, I just let you know that I'm working with a method called functional magnetic resonance imaging, short fMRI. And I'm sure you've seen pretty pictures in the media about it already, where you see like these wonderful blobs. But what it does, it measures the brain metabolism, and this serves for us as a proxy of neural activity. So in fact, we're not really measuring where neurons fire, but where they basically use oxygen, and that's what we're measuring. And what I get, the data I deal with, is 3D brain images. So obviously our brains are in 3D, and then I look at them over time. So we get like every 2 seconds or even faster, like snapshots of the whole brain. And then you can look at it at different spatial scales. So it can be a voxel, and a voxel is basically just a pixel in 3D. At voxel patterns, you can look at regions, which is like the sum of voxels, or of network of regions, which is like different regions acting together. And this is maybe just a bit about the underlying data I work with and the method I use, because you will hear real-time fMRI today probably a lot, because this is the method I use, which just means that I do analyze the data while my subjects are still lying in the scanner. And I can basically manipulate the experiment they're experiencing. And so to give you like a very broad introduction and to see where I'm coming from and why I think bringing machine learning to neuroscience is important, I just reflect briefly on the aims in cognitive neuroscience. So we are interested in very fundamental questions we want to address about cognition. We want to understand what are the fundamental roles of these distinct brain networks we have, and also maybe how we can modulate or enhance these cognitive processes using brain stimulation techniques or pharmacology. And the standard approach in cognitive neuroscience, though, often involves that you select one or a few task conditions in an experiment, and you test this on a cohort of subjects. So every subject's undergoing the same kind of task. And then you draw a group level inference, so very classic statistics, where you then draw an inference about which brain regions are selective for a certain condition you exposed your subjects to. But this is problematic because, as I said, we're interested in very broad research questions, but on the other side, we're using like a relatively narrow experimental framework where we can only test like one cognitive task at a time. And this led to problems in the field, such as that we often come with this method to an over-specified inference about functional anatomical mappings. And with this, I mean that we think that we put an overly narrow role onto brain regions or network of regions. And this is because we only tested a certain cognitive task in this experiment. So we cannot draw inferences about any other cognitive task we haven't tested in that experiment. It also is a problem when you think about patient studies. And for us as neuroscientists, we're also very interested in trying to detect biomarkers. This means that there is like an end aim in the way that we want to, at some point, look into a brain of somebody and diagnose them. Or, for example, predict how they would respond to treatment. But the problem is that how we pursue biomarker discovery research now with fMRI is that we again exposing patients and controls, healthy controls, to different cognitive tasks. And then we want to see where the brain regions differ in response to them, because this could give us an insight about what kind of brain circuits are disrupted. But again, the problem is that we ad hoc usually decide what kind of tasks to use. And without testing all the tasks out there, all cognitive processes out there, it is difficult to really understand whether we found a very sensitive biomarker and whether there are maybe more sensitive biomarkers out there. And then the same goes for non-invasive brain stimulation. So those are techniques where we're applying electricity to the brain in order trying to modulate cognitive processes. And here you can play around with like a lot of free parameters, such as different frequency, different where you stimulate, like on which parts of the brain. So here the problem is that, again, in this kind of standard approach in cognitive neuroscience, you would decide before an experiment starts what exact frequency to choose. And this can lead to confusions around the efficacy of those methods, because due to differences in anatomy or also pathologies, these decisions may actually also differ across different patients. So the only thing I think you need to take away from this is that the standard approach has limitations in cognitive neuroscience. And that's basically where my method comes in. Because we had the idea to not limit ourselves to these few experimental conditions a priori. Instead, we're starting with a broad experiment space and iteratively find an optimal experimental condition. So this was the first study we conducted using this new framework, and it's very much like a proof of concept study. This is not in itself an interesting cognitive question, but I will come to this later. But this is an experiment space that consists of different complex visual and auditory stimuli. So we put them together as audiovisual stimuli. And we ask our algorithm to search through that space without any knowledge about what kind of conditions would evoke what kind of brain response. And we ask the algorithm to iteratively try to maximize activity in the visual cortex, in the higher level visual cortex, and decrease activity in the auditory cortex. And we know just from many decades of research that visual complex scenery would increase and maximize this, this activity in the visual cortex, and obviously no sound would decrease this. So this was basically a proof of concept where we had a ground truth. And that's where we first tested our framework. And the idea is to, for the framework to pick out different experimental conditions and expose subsequently subjects to these experimental conditions. And then in real time analyze how the brain responds. And then based on this information, search through that space intelligently and sampling that space. And we use a machine learning method called Bayesian optimization for doing that. And it will become hopefully much in a second. So what I want to talk with you about today is I want to give you like a brief algorithmic introduction into Bayesian optimization. Then, because I heard it's like a very interactive workshop, I have a little quiz prepared on Kahoot. I'm not sure if you know the platform. Then I present one study where I applied the framework in action, actually in a stroke patient cohort. And then we can, I think, have a bit of an interactive Q&A. And if time allows, we can also reflect on how the framework may have implications for the reproducibility crisis we face in cognitive neuroscience and also other fields of science more broadly. And then again, we can have a discussion if time allows. Briefly start with an algorithmic introduction into Bayesian optimization. So just to place it into the right kind of taxonomy also in your head, it is, in this case, I would put it into the framework of neuroadaptive paradigms that are basically paradigms that adapt to the neural state, so the brain state of the subject. It's a closed loop approach because the algorithm by itself samples and completely controls it. And obviously we're using machine learning, and it's part of a supervised machine learning technique. And importantly, it's part of an active learning where the algorithm identifies its own training data. And that is very convenient because using fMRI and having human subjects lying in the scanner, we cannot exhaustively sample the whole experiment space. I think that's important to mention. Like, we only have a limited amount of time where subjects are basically awake and not fatigued, and that they can involve in cognitive tasks. And fMRI scanning is insanely expensive, so you also cannot just keep them there forever. So we need to make sure that we are unfortunately in the situation where we cannot have loads of training data. And that's why we have this active learning approach, where the algorithm identifies the training samples by itself it wants to learn from, and then we sample, and then it updates itself. So now I already kind of pre-shadowed a bit on the actual approach. So it's a two-stage procedure. And in the first stage procedure, we're using a Gaussian process regression. So basically to update our knowledge about the subject's brain response across these experimental conditions. And in the second stage, we have another function that closes the loop and proposes where to sample next. So that's this kind of intelligent search it will do. And in the first stage, what we do is we choose a covariance function to start with. So that's kind of our prior we put in, and that's why it's also called Bayesian optimization. And we often use the squared exponential kernel, but you can use also other covariance kernels depending on your research question. Just as a brief intuition, so the squared exponential kernel is basically just the Euclidean distance between two experimental conditions in that experiment space. And what it simply means is that we're assuming smoothness, meaning that we're expecting that the brain responds similar to experimental conditions that are close to each other in that space. And that's a very valid assumption for many of our problems, right? So let's go through a quick example. So this is like now a 1D example where we have our experiment space on X. So you can imagine we have different experimental conditions here. So let's say an easy visual stimulus, and then a more complex one, and then a very complex video or so. And then we have our fMRI signal. This just means how much does the brain respond to that stimulus? How much does it basically activate? And by choosing the covariance function, we're basically choosing many different possible like functions to begin with. So these are our priors. So we have different covariance functions that would be possible before we observe any data. So again, this is the prior we put in. Then we start our experiment. We're putting our subject into the fMRI or into the MRI and conduct functional magnetic resonance imaging. And in this case, now expose subjects to these two experimental conditions. So let's say less complex stimuli, more complex stimuli. And now we're fitting our Gaussian process regression across space. And as you see, obviously we need to fit the points we measured. And now we see the space of possible covariance function shrinks because they need to fit the points. And the nice thing about this approach is that we're getting the mean predicted activation of the brain across the whole space, even though we just sampled from 2 points. And we're also getting the uncertainty of the algorithm, where we don't know much about the brain yet. And this is like a very important information that is being leveraged in the second stage of the algorithm. That is where we propose a new stimuli combination. And you do this by choosing an acquisition function, a so-called acquisition function. And again, there are different acquisition functions available. We often use the expected improvement acquisition function, but again, this is just one, and you can trade off exploitation, exploration by the choice acquisition function. And I want to again guide you through this intuitive 1D example. Again, the setup is the same. The experiment space is on X and the fMRI signal on Y. So now what we've seen before is the Gaussian process regression, right? Which in this case is now in blue here. We have the points we sampled, or the experimental conditions we sampled as black dots. And then the true objective function is the black dashed line. That's how the brain is truly responding to all those stimuli, but this is unknown to us. And we would like to model this response. And that's why often Gaussian process regression is also called a surrogate model, because we're trying to model the function. So now to come back to the acquisition function. So we're now also computing for each point in space the so-called acquisition function, and we're maximizing the acquisition function. So in this case, with the expected improvement acquisition function, we're going to sample next where we have the highest predicted mean, which is here, plus the highest expected improvement, so the highest uncertainty towards the maximum, right? And this is where our algorithm will sample next. So this means now the subject will be exposed to the experimental condition that lies here. And then we're showing this to the subject, and then we're sampling their brain again. And now we're refitting the Gaussian process regression. We're recomputing the acquisition function. And now this is the point where we sample next from. And a really nice feature, again, of Bayesian optimization in general is that it kind of transitions from a more explorative search, where it often tries to decrease uncertainty, to a more exploitative search, where it will finally sample around the maximum. You get a very good impression of the maximum of the objective function in this case. So just to show you how this really looks, we have this little video, which is actually from a paper with healthy participants. So again, as I said, we're putting subjects on our scanner. And in this case, we have a 4x4, like again, a 2D experiment space. And now we're actually optimizing across cognitive tasks. So that's a much more interesting experiment for us. So we're exposing the subject to the task, and now we're measuring the brain activity in response to that in certain networks we are interested in. In this case, we get not a really high response. Again, remember, this is an optimization problem. We're trying to find the optimum. Now we're moving into the space, in this experiment space, to the next experimental condition, encoding, which is a memory task. We're showing this to the subject. It has to remember like for 30 seconds or something at something. And now you see this is much more optimal. So what the algorithm will do over time is to converge onto the optimum and sample around the optimum to identify regions that are more optimal, so to say. And the efficiency really comes from the approach that it will disregard regions of the experiment space that are suboptimal for achieving its optimization aim, and to then move onto regions where we identify an optimum or very close to an optimum. So now I have the quiz for you. So for this, wait 1 second, I would like to use the platform Kahoot. I don't know if you know that, but you just need to basically go to Kahoot it and then enter this game pin here. And then we can play this together, and I also see the responses, and I used this in teaching before and I quite enjoyed it. So I will see if people logged in. So again, you have to go to Kahoot it. Let me know if there are any problems with that. Okay, cool, people are coming in. So from what I saw in Zoom, we're like around 60, right? I mean, I'm obviously not thinking that everybody will come in. And there's also this lovely music here. I'm not sure if you hear that. So I feel how fast this goes, you probably already have used Kahoot before. So let's wait a little bit longer. All right, I would suggest we're starting, and people I think can keep joining. And so these are 5 questions now, and you just have to say whether it's true or false. So the first one is, they make it very exciting. Bayesian optimization aims to find the maximum of an unknown objective function. True or false? Yay! Very good. That was an easy one, and I'm glad you listened. That also was a good feedback to me. Next one. Okay, cool. Now we got like our ranking here. Next question. Bayesian optimization can only model linear relationships between experimental conditions and the brain. True or false? Okay, I see you all been very quick. False, that's correct. It is like the correct response is false because it can also model nonlinear relationships. Well done. All right. Bayesian optimization is useful for objective functions that are difficult and costly to evaluate. True or false? You're too quick for the platform. True. This is exactly correct. We're working with brain data in that case that is costly to evaluate due to many different reasons. All right. Friendly Fox is leading. And the fourth question, Bayesian optimization gains a good understanding of the minimum of the objective function. True or false? I also see that a few more people joined, 123. True. This is actually false if we're basically assuming that we're trying to achieve, that we're trying to understand the maximum of the acquisition function, because it won't sample from the minimum in that case. It will sample more around the maximum. So for me, often, if I'm interested also in the reverse contrast, I need to rerun the experiment and then really optimize for that. You can get a good, like you get a prediction of the minimum, but it's not well sampled. Let's say it like this. So, all right, cool. Then last question. To do experiments with Bayesian optimization, I can randomly arrange my experimental conditions in the experiment space. And now I'm curious. True. The correct response is false. And this is because we're using Gaussian process regression and we're having these priors that assume smoothness in our case. So you can't randomly arrange your experimental conditions in the experiment space. And that's actually like one of the major thoughts you need to have when you want to apply this technique. And for example, if we use cognitive tasks, we before use meta-analysis or different analysis techniques to arrange the tasks so that basically the tasks that respond, where the brain activates similarly, will be grouped together, because otherwise you can't bring in this this prior assumption, right? All right, and now it's very exciting. The third place to experts quit. Second place to funny old specs. And then the winner is Spatial Guana. Well done. 5 out of 5. Perfect. All right. So I hope you enjoyed that as much as I did when I used Kahoot in the past. It's so difficult with these formats because I don't really get feedback, but I saw that you played it. So cool. So now I want to give you an impression of how we actually use the technique. So there was like a lot of theory now, and this is a very recent study I've done in patients. And this is a study because we're interested in stroke and also trying to find predictors of how we can, what kind of therapies we can give to stroke patients. And this is motivated by the fact that post-stroke cognitive and linguistic impairments are limited. So with linguistic impairments, I mean that like many stroke patients suffer from aphasia, where they cannot comprehend or cannot basically also use words to describe things. And cognitive impairments is that they're also having limitations in working memory, attention span, and these kind of things. And most therapies, they're very focusing on motor rehabilitation, which obviously is very important too, but patients also suffer from cognitive and linguistic impairments. The problem is that stroke is a very heterogeneous condition. No stroke is the same. So the functional deficits that come from these kind of anatomical lesions, if you want to say, they are very different. There's no one stroke patient. So there is really like a biomarker to make patient-specific predictions about the recovery and to guide individualized treatment planning. And from the literature, we know that so-called domain general networks play a critical role in stroke recovery. So domain general networks are different from domain specific networks, and domain specific networks are networks that, for example, just involved in language, or motor networks. Those are domain specific networks. Domain general networks are networks that are more involved in processes such as attention, memory, reasoning. So they play basically a role across different domains, and they also interact with these domain-specific brain networks. One of them is called frontoparietal, but you don't need to know that necessarily. The problem, though, is that those networks are much harder to study because you cannot just use one task to understand how this network is disrupted. So how do you characterize how much function they still have in that network? This is very tricky. There's no single task available. You can't, you have to decide before then which task to select. And this is tricky because, again, I said, if you run a patient study, no stroke patient will be the same. So maybe you will select a task that is good for 20% of your patients and not for the rest of the 80%. On the other side, you cannot choose all the possible tasks out there because prolonged scanning is not tolerated by patients. They are getting very fatigued easily, stroke patients in general. And they're also their attention span is limited. So what we did is we're running this AI Neuroscientist study, which we call officially Neuroadaptive Bayesian optimization, where we spend an experiment space consisting of 7 different cognitive and linguistic tasks. So those are tasks that were chosen that we know that engage that network to a certain extent, and also across different task difficulties. So we're searching through that space in real time. And here we aligned these tasks based on pilot data. So we had some healthy controls being collected before. We analyzed how similar are basically the brain response to these tasks, and then we ordered them. And now what we did is we conducted this study in 11 patients and 14 healthy controls that are age matched. And the first question was, is this technique feasible for patients? And yes, you don't need to understand the specifics here, but the important thing is patients are able to perform multiple tasks on the scanner. So this was the first worry, because it is cognitively quite engaging to learn these kind of 7 different tasks and keep in mind what the task instructions were. Then also patient-specific profiles are reliable. So what we always do is we conduct 2 independent experimental runs where we completely reinitialize the algorithm and then basically just spatially correlate the predictions we obtained from the Bayesian optimization algorithm. And you can see that while controls have a higher reliability, still patients have a significant reliable profiles. And also often a concern for cognitive neuroscientists is that they're not moving too much, because movement, obviously in the MRI scanner, will give you a lot of challenges where you measure in the brain, and also give you spikes and spurious activity. So that's important. So now what I show you is the prediction of the Gaussian process regression for all our controls. So those are, again, healthy people. And so this is the different tasks on X and task difficulty on Y. And what you see is that our tasks or our controls have a highly similar profile of these domain general networks across the task space. And this is because we don't assume too much differences across healthy people, they all show that they activate most strongly for like a calculation and synonym judgment and encoding task on the highest difficulty level. So this is very predicted in a way and also aligns well with my previous work. However, when we look at patients' profiles, we see that they really all have individual profiles or idiosyncratic profiles of network dysfunction. So this gives us an idea on which kind of task domains patients are basically disrupted, but also where they still have residual activity. So this is maybe also important to mention that we could measure them, and we have measured them behaviorally, but maybe behaviorally, they actually show a really, really bad response. But what we can do with fMRI is to see whether their brain network is still engaging. So we think that this could be a good potential way to then target treatment to. If we find that they're still activating, that they're still having some engagement to that task, that we could use that to restore that network. Well, for example, we could ignore tasks where they have like no response, for example. What we also did is we basically pairwise compared the similarity of all those profiles and projected them back into 2D space using multidimensional scaling. And here you see that controls cluster very similar together, and patients really lie around that in a multivariate distribution. This also shows that no patient is similar to each other, like the patients in general are not similar to each other. You can't really say this is a stroke cohort, this is a control cohort, but patients, they are very unique. So, and interestingly also, but this obviously you can't take, this is a very explorative finding because this is a correlation across just a few people, but we looked at the similarity of patients' profiles, and we saw that they also relate to task performance in the scanner. So even when accounting for lesion volume. And I will summarize what this means because maybe this is a bit confusing in the next page. So we do think that we could use this kind of method for tailoring rehabilitation to individual stroke patients. And first of all, these results show that there's no one size fits all approach due to this great heterogeneity. And I hope I could convince you with this. And then also these patient-specific functional profiles, they carry meaningful information about a patient's behavioral capacity. So we also hope that it carries meaningful information about a patient's future behavioral capacity so that we could use those profiles, for example, to predict how they will rehabilitate. So we think it could guide individualized therapy, for example, by identifying a set of tasks that can be used for training outside of the scanner, or we can also combine it with non-invasive brain stimulation. And this is like also work I've been doing I've worked on before. And I think it's probably a good time for Q&A now. And I doubt we have time to go into this, but I don't think it's very interesting or like, yeah. So feel free right now to ask questions if you want.
Yeah, so thank you so much, Romy. I found it really interesting. I also thought the Kahoot was really fun. It also helped to make sure that we all understood the presentation, which is really fantastic. So we have a question. Whoops.
Sorry.
We have no problem. We have a question from Yuhan. Could you explain more how the experimental space was defined and calculated?
Yeah, so I think the experimental space very much differs across my different research questions. And I showed you now a few examples. Maybe let's go to the stroke study because it's the most concrete example I gave you. In this case, I choose together with a clinician who works with stroke patients, cognitive tasks where we know that patients will have difficulties on, and cognitive tasks that we know though that patients still can do. So we don't want to use like too difficult conditions where they get frustrated or where they would get too tired. And then we chose these tasks because some of them are linguistic. So we have ovid naming, semantic judgment, and so on. And some of them are cognitive, like calculation and encoding. And in this case, what we've done is we exposed like a pilot, we conducted a pilot study where we exposed healthy controls to all these different tasks in various different ways. And then we run like similarity analysis between those, and then order these tasks so that the tasks that that basically elicit a similar response in these brain networks we were interested in would be grouped together. And this is really that we are not violating this Gaussian process regression assumption. For other like this is very much the case for cognitive tasks, it's much more challenging to sort them. For other experimental conditions, for example, when you deal with audiovisual stimuli that you order according to the visual complexity, it's a natural dimension you would choose. There's nothing you really need to think of. The same goes for brain stimulation dimensions. If you search through this kind of experiment space, there's an intrinsic ordering already in there where you don't have to put so much thought in. So it very much depends, but the importance is that that you come up with the experiment space, you decide what you put in. So in that end, obviously the researcher is still very much involved, because sometimes we call this the AI neuroscientist. And also the ordering you take care of. And sometimes it can be harder and you need to use heuristics, as I explained with the cognitive task. And sometimes it's more natural and just comes from the experiment you choose. For example, task difficulty, again, is an intrinsic dimension. There's nothing you need to think of. Obviously, you will expect it hard and medium is closer to each other than hard and easy, right? Like in terms of brain response. Right. I hope that answered it a bit.
Absolutely. Well, another thing I was thinking about is I'm sure you use the stroke recovery example, which was great to go through. I can imagine sort of a lot of other application areas as well, Parkinson's, maybe mental illness. I was wondering, when you think about the possibilities, what excites you most about the possibilities?
I think what excites me most is really the focus on the individual, which is very rare. I mean, I have to say in cognitive neuroscience now, the trend in general goes a bit more onto focusing on individual subjects and trying to understand them very well, but it's still kind of a new avenue. And it's, but I think that excites me, especially with respect to also mental illness, because there is also in psychiatry and in cognitive neuroscience and clinical neuroscience, this understanding that the diagnoses we give people, they're kind of arbitrary. They're based on certain decision criteria. So if we're focusing on individuals, we can also very much focus on what is actually happening in their brain. And maybe this diagnosis we gave them is not entirely correct. For example, there are certain psychiatric conditions that you could, that are on certain symptoms are much closer to each other than on others. So you could think of completely reordering how patients being treated or how they're being thought of in terms of groupings.
Right, I can imagine that. And I can imagine how that would also help clinicians who would be able to look at that data and then adjust medications to better help each of the patients. I can imagine how that would help an enormous amount.
Yeah.
So I see a question from Sujin Lee. She said, first, thank you very much for the excellent presentation. And she would very much like to implement the neuroadaptive Bayesian optimization algorithm in her brain stimulation experiments. She said she just doesn't know where she should start in terms of coding the algorithm. Is there any resource that provides MATLAB Python codes that I can have a look at and modify as necessary from my experiments? So Sujin.
That's a great question. Yeah, maybe I should have put it in there. So in my brain paper, wait, I put the reference up again. Here. There is a link to a GitHub where I put all the code in. And this is the code also to the real-time. So all the real-time fMRI code is also in there. So basically how you would do the motion correction real time and so on. And also the Python. There's also Python for the Bayesian optimization part. But there are there are many different Python packages for doing Bayesian optimization. You can use the one we uploaded, you can use other ones. Also MATLAB has inbuilt tools. So to leverage Bayesian optimization by itself is I think straightforward using different techniques, but then to link it into these kind of experiments is a bit more tricky, I think. And for brain stimulation, I actually have another paper where we used, there's also a GitHub in the paper. But this is a GitHub which is not by me, but by somebody else. So, but yeah, so you could start there and also always get in touch if you have more questions or more concrete problems you're encountering or so.